The reason I'm here is not because I elected to be, but because I was told to be by Sue Landgraf. And since I kind of got used to living a little bit longer, I certainly didn't want to say no. And especially if you put Sue Landgraf together with Siri, you've got a combination that could defeat and conquer Joan of Arc. So that's why I'm here, ladies and gentlemen. But I've known you 40 years, 4 decades. And it's as shocking as it is surprising. I'm still here talking to you guys. But I would like to say that I'll leave you with a thought that nothing comes from nothing. Everything is connected. One thing precedes the next, precedes the next. This word precedes the word now. This breath precedes this breath now. This thought precedes this thought. And on we go from one memorized or moment of memory to the next. So everything is built upon something. Nothing comes from nothing. And CFRI came from these people, none of whom I ever knew, but they formed this organization in 1975. And then 2 years later, they arranged an award, and I was the one to receive that first award. So I guess that has something to do with why I'm here too. Now, when Sue gave me this task, she said, "Oh, we're going to help you because we'll give you the list of the things that we've done, and we would like for you to talk about the significant research that CFRI has been involved in awarding for the last 40 years." Yes. I really should have said no, but I didn't have the guts. I certainly don't have the intellect or the experience to cover as much as this organization has covered. And let me just give you an idea, because I took this sheet, this Excel sheet, in which there were any number of awards to individuals. There were something like, I believe, 250 or 60 awards. I'll have that on the next slide, but I separated them into different categories. And I came up with 10 different categories, and you see some of them here. You don't need to read everything there, but somebody else would probably put them in different categories: immunology, sweat, bacteria, gene therapy, mucus, and others, the airways, tissue culture, CFTR, clinical biomarkers. And how am I supposed to tell you what is significant of all that? Because I can't. I don't have that kind of a judgment or the authority. I can tell you, though, that everything is linked to and built upon everything else. And I don't think that any of this is insignificant. So in trying to get around this or get on top of this issue, this enormous thing, I started noticing that in the list of awardees and awards over the years, starting in 1977, going to present, that there were some individuals whose name continued to appear. And it struck me that this organization has been very instrumental in forming careers in research in cystic fibrosis. And so if I put those together, in those 40 years, CFRI gave something like 259 awards since 1977. And if I look at the first 20 years, from 1977 to 1997, I found that of 42 awards I would consider that 22 of those awardees are still, or have dedicated their life to work in cystic fibrosis, still active or they've passed away. So I think that's an incredible record. We have built here in California a community of people that are truly dedicated to working on this disease, and CFRI was core to forming that community. So before I go on, I'd just like to remember someone that is very dear to us and contributed an awful lot. Dieter Grunert. Dieter passed away last year of a sudden heart attack. But Dieter was a real pioneer in tissue culture, and he identified the activities of CFTR in tissue culture. He contributed much to developing an immortalized cell line. What that means is cells that just grow and grow and grow without dying, which allows you to continue experiments more or less indefinitely. He was very much concerned with developing appropriate vectors for gene therapy whenever we were really on that train. And he recently became very much interested in this new technique, or new science of gene editing. That is a new form of gene therapy in which we don't put another one in, we just fix the one that's there. So Dieter was a pioneer, an innovator, and a great colleague. And I'd really like to just pay tribute to Dieter. All right, so let's get on and see how badly I can bore you with the rest of this. This, I guess, is where my life started in cystic fibrosis. What you see here is, if I can find my pointer, where's my pointer? There it is. Does it show over there? You see it? How come I can't see it? And my computer's about to take off too, so. Anyway. Oh, it's a tiny thing over there. Yes. Oh, I'm sorry about that. Okay, but anyway, I'm pointing to the tip of a pipette. And there are actually 3 pipettes in there, 1, 2, and 3. That's one right there. The second one is outside it, and then there's a third one outside of all that. And that is what we used or built and constructed to be able to pick up the end of an isolated sweat gland or sweat duct, stick that little tube down inside there, and measure things. And below there you see the actual photograph of a native sweat duct, isolated, that's being perfused with a Ringer solution so that we can measure the voltages or the electrical phenomena going on in this epithelium. And as I said, nothing comes from nothing. And that little tip there that you see, the sweat duct is about maybe a fifth of the diameter of a hair, so it's pretty small. That little tiny thing goes on the end of all that. Okay. So this sweat duct is down here on the very tip of, well, this thing is not there, right on the tip of that. So the sweat duct is now built on top of this, but this was built several years earlier at the NIH by Jack Orloff, Bird Grantham, Maurice Berg, and Jared Grantham. They put all this stuff together so that I didn't have to start from the very beginning and build it up. But this is what it takes. So one thing sits on top of another thing, on top of another thing, which is beautifully illustrated here mechanically, but it's also ideologically and with respect to the development of ideas. So none of us can make independent claim to anything, I think. It's all connected. We all depend and stand upon the shoulders of someone else. Okay, this guy. Anybody heard of him? And it is spelled W-I-N-E, like what you drink. And certainly he's given me plenty to drink over the years. But in many ways. Jeff's a very dear friend of mine, really. And the reason he's a dear friend, because you probably don't know this, but Jeff was the first person that understood what we were talking about whenever we said chloride impermeability. I had a hell of a time with this whenever we came out with it, because in those days, the organization was the CF Club. And the CF Club was about 100 clinicians who got together once a year and talked about the clinical problems with cystic fibrosis patients. None of them were physiologists. None of them really understood electricity. And when I got up and talked about this stuff, it was just like going over the moon. So I was standing in the kitchen of my home one evening. The phone rings. I take it off the wall, and there's this guy says he's Jeff Wine from Stanford. And then he's just read my paper, and he started talking about it, and I thought, hallelujah, somebody out there got it. And it was Jeff. And I guess it's okay to say that I guess everybody knows that Jeff's daughter had just been born with cystic fibrosis. And so he started reading, and that was the beginning of a friendship that has lasted more than 40 years, or about 40 years, something like that. Many years. My dementia's acting up again. So, but Jeff has been an incredible contributor to our community as well. And this little inset that you see is a tribute to his innovation, because what you see are a bunch of little sweat glands popping up with blue dots on top of them that he innovatively added blue dye to so we could see them. And Jeff began with an interest in hyposecretion of sweat glands. And we know that that's controlled by a kind of stimulation called beta-adrenergic, and it's very defective in CF. So he started that, and he's honed it over the years to something that's really a very admirable and sophisticated technique for determining just how much CFTR is active in the native sweat gland in vivo, that is, in the human body. You don't have to cut it out and do all those sorts of things. You can measure it just as it is, which is a great advance, because now we have the potential for measuring the effects not only of existing CFTR, but of treated CFTR with the drugs that are now becoming available. It's a terrific contribution. A couple of other things that Jeff did. He got his fingers into several pies along the way, but he became something of a patch clamper, but he gave that off to other people in the lab. And then he developed this thing that was a virtual gland. And I just can't help but admire that, because he took tissue culture cells, mounted them, and then made this little thing with a tiny hole in it so that the cells would secrete into the hole, and then he could measure the volume of these cells actually secreting. So his recent work has been to show that CFTR, just a little bit of CFTR is very helpful, because we now know from his work that you don't have to have but a few % of the normal activity of CFTR to survive fairly well with this disease. So what else can I tell about you, Jeff? I can tell one more thing. This guy scared the hell out of me. We went to a conference in Seoul, and we left Seoul, and we got over the Pacific Ocean, and I look over at him, and he's as pale as a sheet. He looks like a clammy glob, and he says, "Well, I don't feel so good." And I look at him, and holy cow. Well, I thought he was going into a cardiac. I think he thought so too. We called the stewardess and asked for help, and there wasn't a single physician on the plane who responded. So fortunately, I got to questioning him and asking him if he did anything. He said, "Well, I took a couple of doses of propranolol to keep my blood pressure down." So he poisoned himself before he got on the airplane. And I had to decide whether we were going to go back to Seoul or to San Francisco. So when I found out that he did that, I said, "San Francisco. If you die, you deserve it." What a friend am I, huh? Oh. What about this guy? Rick Boss. Rick has been around for Rick's our resident immunologist. So he's been interested in, pardon me, immune complexes, especially with respect to hormone responses and Pseudomonas infections. Maybe I can see this a little better if I get out a pair of glasses at work. Nope, not those. Oh, that's better. Yeah. So anyway, so he became very interested in immunology and cystic fibrosis in the early 80s. Continued that line of work into studying hypogammaglobulinemia. And to me, when I think of Rick, I have this image of this guy going with my wife and Norm Lewiston down to the Primate Institute in Davis to use monkeys in those days as a model for cystic fibrosis, studying cystic fibrosis in animals. So he was doing endoscopy on monkeys. And I guess he decided that was kind of too expensive because monkeys, well, they cost a lot. But anyway, he continued his interest in immunology and has continued that into the present day, in which he's now identified a biomarker that indicates the presence of aspergillosis. I'd also point out two other things there, that he was one of the first people to start a comprehensive mutation screening program in the CF centers, which we all know we desperately need, and has certainly filled a wonderful, it certainly filled a real gap that we desperately need. The other thing I put down here, which is kind of a thought thing, that is pitfalls of drug development and lessons learned from trials of donepezil. And it's just kind of an editorial thing, but it gives an incredible amount of insight from somebody who really got into and kind of understands the pitfalls and the problems of developing a drug. And it's a great paper to read because it's a warning about getting too excited too fast. Not to go so fast that you can't stop before you fall over the edge. Anyway, Rick I think has really contributed to our community for 4 decades. Jonathan Whitcomb. Whitcomb preceded me in retiring, but he certainly left his mark on our community and dedicated his entire research career to work in cystic fibrosis. Jonathan was largely concerned with the airway surface liquid layer that covered, or the liquid fluid, the fluid that covered the airways here. In that upper right corner, you see one of his frieze etchings of the airway surface mucus here, and then with what he calls the SAW layer, labeled by S down below it, with cilia beneath that. And he spent a lot of time trying to figure out why this mucus in CF found in cystic fibrosis lungs doesn't move. For many, many years we thought, well, it was just dehydrated and it got stuck because it didn't have enough water, and it pushed the cilia down so they couldn't move, and there we were stuck. But Jonathan spent a lot of time on tracheal epithelias in cultures. He helped Dieter Grunert and Walt Finkbeiner in developing culture systems for study. They got them so that they could carry them for long terms during study. And he helped with transforming human cells into cells that were essentially immortal. His primary love, though, was the regulation of the airway surface liquid. How do you make it bigger, littler, thinner, thicker? What controlled that? And that he carried into trying to figure out the relation between mucus and secretion, chloride secretion. And lastly, just his, maybe it's his swan song, but he and Jeff Wine got together and wrote an incredible review on the airway submucosal glands. And if there's anything you want to know about submucosal glands in the airways, take that paper. It's a wonderful contribution. All right, Alan Verkman. Alan got his first award in 1987, and I think if we have an Einstein in the CF community, it's Alan Verkman. This guy finished his public schooling about 3 years ahead of everybody else. Came to San Francisco, set up a lab, and he's the guy who came up with the idea of using a chloride-sensitive dye to study the activity of CFTR in cultured cells. And what you see here is one of his pictures of a dye that's called SPQ on the left and another called LZQ on the right. But the intensity of those dyes, the fluorescence, is proportional or inversely proportional to the amount of chloride that's in the cell. So if you activate CFTR and chloride is going through it, it will either leak in or leak out much more rapidly, and you can measure that as a function of the channel activity by measuring the intensity of these dyes. It's been an enormous contribution to the field, and in fact, it's the basis of all the screening that's been done to bring us the Vertex drugs in the last couple of years. So Alan's contribution is immeasurable in that regard. He was also responsible for pushing and developing these compounds with what we call high throughput screening, which is a machine that takes these cultures and just zip, 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 zip like a robot and says this one's good, that one's bad, and this one's something else. So that is the basis, again, of the drugs that we now have that are treating cystic fibrosis. High throughput screening, looking at changes in chloride in the chloride indicator fluorescence for activity. Another thing that he did for us it's really an enormous contribution is developing potentiators and activators and inhibitors of CFTR. Again, right back to where we are now with these drugs that are being used therapeutically. So, and he did a number of studies looking at ASL composition and depth and viscosity. So, to our Einstein, Alan Verkman. Phyllis Gardner, she got her award 1988. And I don't know Phyllis very well, but only sort of by reputation. And then when she came out with this finding, I thought there was going to be a civil war in Northern California, because no one wanted to believe that CFTR was anywhere except in epithelia. And she said, no, here I found the CFTR in lymphocytes. So why is CFTR in lymphocytes, and what problems do we have with lymphocytes? Because really the only place that CF patients get infected is the lungs. You've got lymphocytes everywhere. But in any case, there is CFTR in the lymphocytes, and she went on to show that you could stimulate that with what we call P1Y receptors, or agonist, and that's a different form of what we usually use to stimulate CFTR, but in any case, she contributed that. She's also very interested in gene therapy and developed some methods of transfecting cells with normal genes. She used the maxillary sinuses, which she liked to focus on. And then more recently, she's been developing a genotyping microarrays to help us, microarrays for helping us to identify the different mutations that occur in cystic fibrosis. 1990, Judy Palmer received her first award, and I was quite surprised that she began with an interest in developing a sweat test, which is what that NSAID is about. It's a device for, or it was papers that she used to determine or to get an idea of how much chloride there is in the sweat. So based upon what the reaction she got to that. But she didn't follow that very long. There were lots of people going after sweat tests. Her real interest, I think, was in meconium and meconium screening, and it continued to be screening in the newborns. She also was one of the people that became interested in pregnancy and cystic fibrosis and what its ramifications were for young adult CF mothers. She got into the nose with bacterial biofilms, identified that we had biofilms in the sinuses,
And she is very concerned about prenatal screening and therapies that we will apply at the beginning, very, very quickly after birth, once we can identify the presence of the disease early on. And the last thing that I put here, and believe me, I am only putting a smattering of each of these, what each of these people have done. So I'm sure I'm making mistakes and that sort of thing, but I hope to convey to you that this has been really a terrific effort from these investigators who have committed themselves. But this last thing, I think, is a frontier for us that we may want to continue to look at much more closely, especially if you're looking at the future of CFTR and what we're going to be doing. CFRI, I mean, because she found that this genotype, T2R38, correlated with a better quality of life with respect to, should I shut up? A better quality of life in people with Delta F508. And the reason I say that's important is because I am convinced that we have compensatory genes, that we have compensatory mechanisms that can fill in for CFTR whenever it's defective. And if we can learn how to manipulate those when they need to be activated, bring them into play, I think that may be a very, very strategic way to ameliorate the devastating effects of defective CFTR. And that will be particularly important for mutations that do not respond to our small solutes, that do not respond to these vertex compounds. So, Terry Macon. Terry's another old friend of mine. I don't know how we get on these tracks and we just start to stay together through life, but it's kind of wonderful when you get this old. And how many people in here are over 70? Hallelujah. I am so tired of being the oldest one in the grocery store. So anyway, Terry and I, when I first came to California, I came to UCLA to look at doing a postdoc, and that was about 1971. And Terry was in Jared Diamond's lab, and just across the way from where I was looking at a postdoc with John Torby. And Terry had a house in Venice, not a house, a cabin, a bungalow, a shack. And so Terry took me in and put me up while I was staying there. And he was into surfing. I didn't do that. 10 minutes only? Holy cow. Have I really been talking that long? Oh, they shut that thing down for a while. That's right. Okay. All right. I will run on very quickly then. But Terry was a great friend, and we've kind of followed each other over the years. And I really owe him a lot because in the beginning, I was sure that bicarbonate was not permeable through CFTR, that CFTR was impermeable to bicarbonate. And Terry was one of the most influential individuals to change my mind about that. And now, of course, I've become a real prophet of CFTR, an advocate of bicarbonate and CFTR, and I think it's really at the crux of this disease. So, Alan Finkbeiner. Alan is also really an amazing scientist and contributor to our community. And if I would say there's any difference between Southern California and Northern California, it's that you guys in Northern California have Alan Finkbeiner. Yes, because he is one of the few, maybe the only pathologist I know who knows something about physiology. Pathologists are notorious for studying the dead, and they don't care if it's alive or not. And if you as a physiologist go, and physiology is life, no? You want function. It's got to be alive. You ask the pathologist and they hand you this dead stuff. But Walt would give you live stuff. And that's why all these guys here have done so well, because they've been able to get human tissue live that they could study, that they could make cells from, transform them. So Walt's contribution to that area is, I think, unparalleled. He also introduced the Cal-U3 cell and studied submucosal gland dysfunction along with Jeff and others, and he was also very concerned about control of mucin secretion. And then more recently, he's gotten in on the wagon now with ivacaftor and its effects in patients with CF. Okay, Doug Conrad, probably not well known to you up here, but he's our center director at UCSD. And in the last few years, Doug has really blossomed. He got his award in 1995. He had so many clinical responsibilities earlier that he kind of got suppressed. But more recently, in the last 10 years, he's gotten interested in lots of things that affect us in cystic fibrosis. I list here sleep disruption, big deal. God, I can't sleep enough. He got into studying viral communities in the CF lungs, which are very important and have been really, I think, terribly neglected, because these guys are the ones that precipitate exacerbations. He worked with us a little bit on beta-adrenergic stimulation, and more recently he's really gotten into this fascinating area of the ecology of bacteria and pathogens in the CF lung. And this beautiful picture, which I selected for its colors, and I don't really understand, reflects this. But basically what it's saying is that if you look at any of these ellipses, the numbers there represent the number of metabolites that are included. And so where the Venn diagram overlaps gives you the number of metabolites that are common to those two overlapping areas. And what he and Forrest Rohrer have found is that the different states of the infection, the CF lung, can be characterized by the metabolites that you isolate from the sputum. So you can characterize when an exacerbation is occurred or about to occur. You can look at the differences that occur after you treat it and get it cleared up. You can see what it's supposed to look like while it's stable and while it's being treated. So this offers us an enormous tool, I think, to evaluate when we're going to have a crisis or an exacerbation and how we should respond to it. Not to mention what we can do by knowing what these metabolites are and if we can control and manipulate these. So I think it's a great place for future work. Horace Fisher. Horace got his award in 1996. Horace is our, I guess, premium patch clamper, but I can't say that with Beata in the room. Because both of them certainly know how to wield a patch clamp. But Horace was one of the first persons to show that CFTR had two doors, or two ways of opening the door. Two gating modes is what it's called. And then he went on to show how you could regulate that. And then most interesting, I don't know how this came about, and I don't know who to put where, because if you guys know Beata and Horst, you know they're married, and I can't separate them. And so I don't know who's the herbalist in that family, but they have a fixation on things that come out of herbs that you might put on CFTR. And they discovered that genistein, which comes from, I can't say the name of the plant, but it's some plant that they got dye from. You can make yellow dye from this thing. And people used it for that. But anyway, genistein affects a phosphatase, and they found that they could activate CFTR with it. And then not only that, but they could correct the G551D mutation with this thing, which was a really a big deal, because then it sort of gave us the foundation for saying that you could do something about these molecules with another small molecule. Horace was also very active in studying acid and base secretion in tissue. He's very fond of pushing forward his idea of a proton pump in these airway cells that contributes to pathology, and Mike Welsh has advanced that theory significantly in the last 5 years, so that the CF lung is not only a problem of not having enough bicarbonate, but a problem of having too much acid. And so the reason you have too much acid, of course, is because you don't have the base to neutralize it. But to move on very quickly, as I said, I can't separate and pull apart Beata and Horst. And so here I violate my rule because I go beyond 1997, because that was when Beata got her first award. But I suspect she had a lot to do with Horst getting his first award. So I think it's only appropriate that Beata be included in these people who have devoted their lives to studying cystic fibrosis. And in fact, again, she was very instrumental, maybe the instrument, in showing that our famous, notorious, outwardly rectifying chloride channel was not the cystic fibrosis chloride channel. And we went through all sorts of agony with that for a number of years. And finally, it was just established that this guy was not doing the bad deed. It was actually CFTR. So, and again, she contributed or probably invented the genistein effect. I don't know which one to lay that blame on, but probably Beata. And she also said that vitamin C can be used. And she's also, of course, interested in other ways of getting chloride in and out of the cell with different types of other channels, calcium activated channels. And I very much appreciate the fact that most recently she's become interested in air pollution, because air pollution for patients with cystic fibrosis is a real demon. And so if we can figure out how to minimize the damage from air pollution, I think we're all going to be, not only us, but you guys too, everybody's going to be much better off. So go for it, Beata. Oh, what is this? Capito, 1960. Oh, what you see here is a study of the thickness of mucus in normal and CF patients by Capito. This guy, this is Lou Capito. You know who Lou Capito is? He's the father of Ron Capito. CFRI has a legacy in Capito because Lou Capito worked with Harry Swakman. People recognize the name Harry Swakman? One of the most renowned and erudite clinicians in cystic fibrosis. He really defined the clinical pathology of this disease in the late 50s and the 60s and 70s. And Lou Capito was a biochemist that worked with him, and he defined the composition of nails, of hair, of skin specimens, of earwax, everything you can imagine that you could get from a CF patient to see what and if is abnormal. And so we have his son here at Stanford who has contributed enormously to CF, in that he was involved in the first cloning of a gene, the BAN3 gene. And then he went on to show that this expression is not correlated with the rectifying chloride channel, which we were just talking about, and that there is a degradation, that we have this, the basic problem with delta F508 is that there's a premature degradation of this protein. And Ron is responsible for showing that. And he then went on to show that you could reverse this by incubating these cells in high glycerol. Again, supporting the idea that small molecules can affect the protein so that we can get to a functional state. Very important. And he went on, of course, more recently is showing that there is this ubiquitin, which is a marker for these degraded proteins or pieces, accumulates in several other disease forms as well. So Ron Capito is our legacy from Lou Capito. I'd like to make a couple of exceptionals here to my rule, because I'd like to say that Julie Desch has really contributed to our community. And she started, I think, she was a medical student and worked with Jeff Wine on hyposecretion in the sweat glands. But then she went on and got a family and then dedicated herself to this this field. And what I really like about this is that, I want to tell you guys this, I get me off on the preaching here and it's going to be bad, but an illness, or let's say, let me put it this way, health is more than the absence of a bacteria. And just because you get rid of all the bacteria or keep everything clean doesn't mean that there aren't going to be some issues and some unhealthy things going on up here. And cystic fibrosis patients now have a lot of that to deal with because of this, our hygiene, quote, regimes, and the things that we do to protect, but at the same time have an enormous impact on our self-image and have a tendency to stigmatize and ostracize. So something that I think we have to continue to keep in mind and try to improve as much as possible, and I think Julie has really been very important in that. And so she has, she's also developed this mindfulness program, which I definitely need. And I really appreciate the work that she's done. And lastly, I'm breaking my rule here because there's this guy named Fred Van Gore, who's the chief scientist at Vertex. And he's been there for decades. He can't have a CFRI award because we don't award industry. But I think that maybe the next, because of his work in developing the compounds that have come out of Vertex and identifying them, I think the next drug that comes out of Vertex should be called the Van Goricaftor. So we have Goricaftor next. Anyway, okay, so let me just have a couple of minutes and leave you with a lesson of physiology, because all of this stuff, for me, condenses in the next couple of slides. Can I do that, or do you guys want to run me off right quick? Yeah? Okay, I'm going to put on another hat, physiology professor, okay? And I'd like to get two things across to you about why the level we've come to understanding this disease, at least the way I think about it. And first, I'm going to tell you that to expand mucins, you need bicarbonate. And you've heard about chloride impermeability, but I think, and I've come to the conclusion now, that the issue in cystic fibrosis is bicarbonate impermeability, not chloride impermeability so much. This is the thing I think that really contributes to the deterioration. And the reason is because if you look at this, I don't know if this thing has decided to run itself, but I'm secreting bicarbonate, and as the bicarbonate is secreted, the mucin is released, and then the mucin, when it combines with the bicarbonate, there's a calcium in there, pulls it out, and these little mucin granules blow up, explode. Now that may not be so impressive to you until you think about what a mucin molecule is. It is one of the most huge, the biggest molecules in all of biology. And it has many, what's this thing, running itself again? I don't know. Anyway, behave yourself. Anyway, so mucin molecules exist inside cells in little tiny packages called mucin granules. And they have to get out and blow up and explode and become expanded so that they're thin enough, the mucus becomes thin enough so that it can be moved along the surface of the airway. And if it doesn't thin like that, if it stays thick and sticky, then it doesn't get out. And then you've got what we call mucoviscidosis. Yeah? So, if you take away the bicarbonate, you get mucoviscidosis. So we just saw here that the granules come out, they combine with bicarbonate combines with the calcium, and it goes boom, explodes in about 2 seconds. In cystic fibrosis, if you don't have that bicarbonate, this is what happens. Okay, we stimulate, and then we activate, but we can't get the bicarbonate out because there's no CFTR, no way to move it there. And then consequently, when we try to secrete the mucus granules, they go out and they kind of start to explode, but they don't make it. And so therefore, we have mucoviscidosis, we have poor mucus transport, and we get sticky small airways and plugs in the lung, which support chronic bacterial infection, inflammation, destruction, eventual loss of respiratory function. So, let me go a step further though. That's the basis of the way I'm beginning to see the basic defect in cystic fibrosis and how we connect it together with the problem in mucus. It's not so much that it's dehydrated, it's really that it cannot expand, or it does not expand as rapidly as it should when it's supposed to. So this is a small airway. If we cut it in cross-section, it's like a tube. You cut it in cross-section, it looks like, where did it go? It looks like that. Okay, it's highly plicated, lots of folds and things. Our big issue is, how do we keep the surface of that thing clean? How do we keep it clean? We've been talking about the airway surface liquid. It sits on the surface of those little plications. And if you cut that open and look at it, it would look like that, with furrows and ridges in between all those plications or pleats. So what we've found in the last couple of years of studying this thing is that the way we normally keep our airways wet is by the cooperation of cells that secrete and cells that absorb. So previously we used to think that, well, all these cells either secreted or absorbed, but it doesn't work like that, we think. So how does it work? Those furrows that I was showing you a moment ago, if we can let them be represented by the blue cells as the furrows and the red cells as the ridges, we find that those cells, the blue cells secrete in the furrows, and the red cells absorb. So what we have is a recycling of fluid within the airway, all along it. And therefore, the airway normally stays wet. It doesn't get too dry, and it doesn't get flooded, because what is secreted is reabsorbed. And so that recycling keeps us wet. Now, if we disturb either one of those things, we can look for problems. Okay, now which of these is more disturbed or is disturbed at all in cystic fibrosis, I can't tell you now, but I'm pretty sure that both of them have some effect, because they all, CFTR is in both of these cell types, the ones that absorb and the ones that secrete. But this is even more complicated by what we just saw, talked about a moment ago. This is the recycling diagram. And here, if we stimulate with calcium, we can stimulate the secretion of bicarbonate. What is bicarbonate? It's your baking soda, you know? So this is a problem. I don't think anybody's going to get rich by introducing sodium bicarbonate, because you can buy 6 months supply of the drug in the grocery store for about $4. And so anyway, we have to secrete the bicarbonate, because when we do that, we have to provide an ambience for the secretion of those mucus granules, if they'll ever come up here. They're not doing it, so anyway, but yeah, there they go. Okay. So the bicarbonate allows the mucus to come out, the mucus goes out into the airway surface liquid, and that provides this coat of fluid that normally carries the debris that we breathe in back out of the lung before we get infected. In cystic fibrosis, we have a problem because we can't move it fast enough. We get a bug in there, it sits, it colonizes, we get inflammation, and the rest is history, as you know. Okay, where to? What next? My favorite poet, Carl Sandburg. I think we have to pursue the idea of bicarbonate secretion and really figure out how we can either replace it or find other mechanisms for inducing its secretion. We clearly need to find out if there are other alternative channels. Cystic fibrosis trafficking and degradation therapeutics is high on the list. Respiratory biomics and pathogen ecology is very high on the list. DNA and RNA editing, as we mentioned, is something that we really need to pursue. Gene therapy, I don't think that's dead yet, and stem cell therapy. And that's just a partial list. Okay, I'm sorry, I didn't realize that this was going to take so much time. I thought I had about 30 minutes to talk, or I'd get it over in about 30 minutes. But I always end my talks with my paramour. And There she is, to wish you well. Isn't she cute? I think she's really nice. My wife is terribly jealous. She says I spend too much time with her. But she's only about half of the picture, because we discovered that while Mike Welsh was kind of making eyes at her, he noticed that she had a wicked stepsister who is what we call low Phyllis. Low Phyllis without bicarbonate makes the pH very high, very low, makes too much acid. And the acid in the airway prevents the immune system from working properly and therefore enhances infection. So I present to you lovely bicarb, and I'd like for you to stay away from low Phyllis, at least being nice to her.